Thank you very much for the nice introduction, and I guess it's better if I talk in the microphone. And for Gordon for inviting me. And artificial intelligence is an interesting thing. My wife keeps on telling me that she will trust this thing when artificial intelligence can pack the dishwasher. So I don't know how far is it from us, but right now we are trying to use this one to check the effectiveness of chemotherapy and the treatment of lung cancer. So the outline of my talk is some background, of course, the clinical need and glycobiology, the methods, the cohort selection, glycomics and the AI pipeline. And we wanted to check the predictive performance, the results, and how can it be interpreted. And of course, some discussion about the clinical relevance. So the study objectives were the primary aim to develop and validate this artificial intelligence model that using the pretreatment and early on treatment of the serum N-glycome to predict the chemotherapy effectiveness in different lung cancer patients, and to identify key glycan features. And we can use that one for utility and check the prospective applications. So lung cancer is the second most common cancer in the world. In the EU, there's more than 300,000 patients per year. In Hungary, interestingly, if you see the right side graphs, it's higher than in Europe or even in global. So I don't know what happened in Hungary. Actually, maybe too many people smoked or something else, but it's really prevalent there. So the available treatments right now, it's like chemotherapy, radiotherapy, immunotherapy, targeted therapies, and surgery, of course. And the 5-year survival rate is around 20-25%. And the major types of the small cell or the non-small cell lung cancers, and they are checking with different systems, the medical history, and the tumor, and the node, and the metastatic parts, and effects on that one. So the most common treatments are first the chemotherapy. It's like 62% of the patients. It's not really helping that much, so the chemotherapy should be changed. And that's the main reason why we started this study, that if we can assess the effectiveness of the actual chemotherapy, what was prescribed first, they are following clinical protocols, so practically they cannot really change the thing in the first place, but on the second part they can. So, and there is a large heterogeneity in the chemotherapy response. So many lung cancers, like the non-small cell or the small cell, have different sensitivities to different chemotherapies, like the platinum-based or other ones. And the problem is the over-treatment, which is toxic, or the under-treatment when we cannot really gain what we wanted to in the first place. And the current predictors are like the clinical stage, the histology, single biomarkers, some of them, and they have limited accuracy and for the individual response to the treatment. So the problem is that several types of chemotherapy is not clear which is effective. Again, they are using the prescribed protocols. So they start with one and go from there, but it takes like up to 5 months to figure out if that particular chemotherapy agent is working or not. So if we can do it much faster, then it will be certainly much better for everybody, for the patient, for the system, and the cost is certainly that serious side effects can be, and the high cost with the unnecessary treatment. So, and considering the fact that only 20% of the chemotherapy treatments are effective, then it's practically a large savings in that way. So, the solution in that case is to find a new method for early screening. So if we got some surrogate markers, what can be used to say that it's okay or not, that would be very, very useful. So again, it would improve patient survival rates, and it will save valuable time and reduce the harmful side effect and the overall cost. So why to use the serum N-glycome? The serum N-glycome, the protein glycosylation is altered in cancer. It's a couple of presentations already talked about that one yesterday, and it will be today as well. And it changes, it reflects the tumor biology and the host immune system as well. So the serum and N-glycome capture the systemic and disease-relevant changes, so it's important to evaluate that, and it's minimally invasive, so we just have to have the blood test, and it can be repeated for monitoring anytime. And why do we have to combine it with AI, or why would we even considering that one to do that? So this glycan data, again, yesterday and it will be today, it's high dimensional and nonlinear. So it's well suited for machine learning, and AI enables this pattern discovery way much better than we are. So we will be checking the electropherograms or chromatograms for the mass specs, the spectrums. We cannot really pick up those very, very tiny changes and tendencies, but AI can pick up. So there's the reason why we have to use it. And there is a potential to produce the individual's probability scores for that one, the risk stratification, decision support, and so forth, if we are using AI-supported data evaluation. So about the cohort and the study design, then the patient population what we used had advanced non-small cell lung cancer. And they were chemotherapy treated, so the first line treat or adjuvant. So the time points, the baseline, we had the pre-chemo and after the first dose, so practical after 1 week, not 5 months, 1 week. And the outcomes with objective response in that case, and what we wanted to have like a progression-free survival, but again, it's after like 6 months, so we have to do it a couple of times until we find the right chemotherapeutic agent, and the cohort type was very heterogeneous. So that's, we had like 33 patients. That's the list. I don't want to really go into the details there, but it lists actually the age, the sex, and the type of cancer, what they have, and so forth. So this was the cohort what we picked for this particular study, and from each patient we had before and after the treatment, and everything was done in triplicates. So the sample processing for the N-glycome measurement, we used the old-fashioned way, meaning that we, I will get into the details just in a moment. So we had the collection of the blood, isolated, and regular storage protocols, did the glycan release, the cleanup workflow. That's the thing, the reason why I'm saying that's the old kind of methodology, because we had like before and after the treatment, and they just went through the regular steps of releasing the glycans, labeling the glycans, and purifying. It is what we don't use anymore, but these experiments were done before we realized that it's better if we don't do the purification because we can lose glycans. But so in this study practically means that it might not be as perfect as it's supposed to be because we use the old protocol with the cleanup steps. And the next, one of the next presentation, Dr. Jarvas will talk about the problems with the cleanup steps, that we can lose things or we can actually just change things just because of that cleanup step with the healing columns or with all of these methodologies. So again, we have to consider that it was made by the old-fashioned way. And we definitely have to repeat it anyway with a larger cohort. Then in that case, we'll be using the new one, in which case we don't need the cleanup steps, so we will see the real thing. So the data processing and feature engineering, it was the next step is like P detection, alignment, and normalization. We use like the internet standards for that one. And we got the biologically interpretable features, like glycan classes, like complex, high mannose, fucosylated, silylated ratios, and so forth. And we combined it with the clinical features if available. It can be, didn't do it yet. So with the AI modeling pipeline, we used the hierarchical approach for problem decomposition because it was a very, very complex problem. So we reduced the problem to 3 binary classification tests of regression, which means that it's good for the patient. So the tumor was actually just shrink, stationary when it doesn't change, or the progression where the chemotherapy didn't have any effect and the tumor was growing afterwards. So it's a problematic. So they're very important, these classification tests. And the algorithms what we use was like tree-based models, like gradient boosting or regularized logistic regression and so forth. And then we tuned the models, so with nested cross-validation, then hyperparameter search. I will be showing those ones. And the interpretation was based on the sharp values, and those are like from the game theory, that it's like positive or negative. It means that it's really we have the effect or not have the effect, and the feature importance and so forth. And the training and test, since we didn't have too many samples, 36 samples, and 3 of each was analyzed, so it's like less than 100 practically. So we had to do this training testing with 80%, 20% ratio to have the coefficient variant really viable in that way. So we did 1,000 runs in that way, just randomly mixing the sample results in 80-20% ratios, and get that in that way a better evaluation. So that's the separation what we got. We identified 21 features in that, and we used a robotic system, an automated liquid handling robotic system, to have the highest precision of the sample preparation. You can see on the right side the reproducibility, and it was very, very high. And we identified these 21 features in a way that we only considered features with a peak area higher than 1% of the percentage, I mean the relative peak area higher than 1%, and it is what we used for the feature selection. So the implementation process was like such. So we had the diagnostic, that lung cancer was diagnosed, and we started the particular type of chemotherapy. We had the sampling before and after, and then we get the results. I mean that they got the results, I mean the doctors in that way, and that it was this onco team decision to go with another chemotherapy agent or just use the agent what they started with. So in this case, the patient definitely had an additional chance to recover if they changed it fast. And the onco team decision was facilitated really by the results what we provided. And of course, it's for the pharmaceutical companies, it's good or bad in that way that maybe some of the therapeutic agent won't be used that much, some of them will be used even more. So that's an important decision in their side to decide actually which one to follow up with. So this is the pie chart demonstration of the data set distribution, but we used in these 3 different classification tasks of stationary, regression, and progression. And this, the binary in this case relates to the type of the classifier to distinguish between positive and negative cases only. So that's the heterogeneity of the sample cohort, the red and the blue. It means that the true or the false results in that way with the 3 different binary classification tasks. And if we check it one by one from the for the individual glycans from 1 to 21, you can what we could get out of this was with the ANOVA test, that like the G6 and the G15 showed really significant differences with all of those, like the stationary, the progressive, or the progression or the regression part. Like the G6, the disparity between the measurements associated with progression and stationary, the p-value was very, very good with 0.02, so it was definitely less than 0.05. And the G15, for example, the p-value was 0.009 associated with regression and progression in that case. So then we started to set up the hyperparameter classifiers in that way for these 5 kinds of classifiers, like the support vector machine, the quadratic discriminant analysis, random forest, the extreme gradient boosting, and the neural networks. And those were on the right side the hyperparameters, but we actually evaluated to be sure that we got the best results for all of those, and then to check which one to be used further in the line to do the analysis. And this was like after 5 tuning the classification models. Those are the numbers what we used after 5 tuning of all of those 5. And then the results to find the best classification method and model performance was performed. Then we got these results, and I hope that it can be readable, but the main point is that with the QDR, in the QDA, we got the best results of those, like it was more than 0.8 the results for regression, stationary, and progression as well. So in this case, the QDA classifier achieved very, very high metric values, and the average area under the curve for the QDA was like higher than 0.82, and the standard deviation is shown just after the numbers. So then we had the ROC curves, and the ROC curves were really, really good in that case after the fine tuning. So after this training and testing, training, testing, training, testing 1,000 times, we got very, very, very good ROC curves. And under the ROC curves, you can see the confusion matrices as well, but with this binary classification test for the categories of the regression, progression, stationary, with the best classification algorithm of QDA, what I just showed previously, then using the sequential forward selection method, we identified very, very good, like for example, out of the 21 features, we identified like 11 for regression. And again, it was after the randomly generated data set of 80-20% with the AUC of better than 0.9. And these were the relevant glycan structure for the binary classification test, which the SFS algorithm, the forward selection algorithm, actually successfully removed all the insignificant ones. So the significant ones for the regression are listed in the first line, for progression the second line, and the stationary on the third line. And you can see that in here, what is the effect of the individual glycans of the prediction. For example, for regression, like the G17 has the highest power. For progression, the G20 had the highest power. That particular structure, or glycan structure, had the highest power of that one. So in this case, the relevant N-glycome is not only varied among the different classification scenarios, but also differed with their contribution. Again, like the G17, G20, and G18 contributed the most for the regression, progression, and stationary. And like, for example, G12 was identified as an independent biomarker in all three cases, and significantly contributing more or less for the accuracy of the model. So in this case, in contrast, it has low influence of the case of regression, but for example, for the other ones, it had higher. So it's always, what we got was very, very important to check these sharp values, and again, is the game theory thing, if it really contributes or not. And then we just use this Bayes-Born plot for the relevant N-glycans for the classifying the disease progression. And interestingly, that the G6, which is like the fourth from the bottom, had very, very good results in that way that, as you see, the red dots are on the left side and the blue dots on the right side. So it means that the sharp value, which is like when it's less than 0, it means that it has no less effect. And if it's in positive region, like in all the blue things here in the positive region, it means that it has a really predictive power for the classification task, like for example for progression. And as a matter of fact, in this case, the progression was really shown with this sharp dependence plot, where the x-axis shows the ratio of the G6 glycan before and after, so it's decreased or increased. And if you can see, if it decreased, the decrease of that one, so it's -2 or -3, then the sharp value really increased, which means that the predictive power increased when we had less and less of that ratio after the treatment. So let's see the clinical implications. So the potential uses of this one is the pre-treatment stratification, and we can switch decision during the treatment, and we can monitor for resistance and so forth. And the great advantages of this blood-based test and repeatable, so it complements, it can be complemented by imaging biomarkers, and other ones, but it still gives us a significant cost savings because we don't have to administer that particular chemotherapy or any other agent for a longer time. And the path to clinic, that is like prospective validation, what we have to use, and of course regulatories and all of other things. The limitations, of course, actually we have limitations as everything else. Like the technical limitation is like the assay standardization, the inter-lab reproducibility, per platform dependence, really depending on which platform is being used for the glycan analysis. And the biological part, the glycome reflects both tumor and host processes, so it's potential confounding by inflammation, comorbidities, or so forth. The study design, so the risk of overfitting, and the small cohorts, what we had right here, it was a quite small cohort, but we have to increase the number of the patients, and of course the regulatory and reimbursement hurdles for diagnostic grade tests. So with the conclusion, I would say that this novel bioanalytical method, what we developed for predicting the effectiveness of chemotherapy, worked pretty well with this particular cohort what we used. The N-glycans were enzymatically cleaved, labeled and analyzed by capillary electrophoresis with laser-induced fluorescence detection. This AI-based binary classification method identified key N-glycomic changes. And the sample size should be increased, of course, as I mentioned, to be able to say more about the predictive power of the individual level of the glycans. So we don't have to check the whole panel, but we can really rely on just individual ones, hopefully. And our data clearly showed with this cohort that the QDA resulted very, very good AUC values above 0.9, which is really excellent, and personal treatment opportunities can improve. Of course, in this case, the lung cancer survival rates. And with that one, I just would like to acknowledge the help of my coworkers in my 2 labs in Hungary, in Debrecen, in Veszprém, and 2 of my colleagues are here. Actually, they will give presentations right after me. And the support what we got actually from Hungary and the university. And I always finish my presentation to remember Professor Csaba Horváth, the inventor of HPLC, which we named our lab after, so there's a reason why it's a Horváth Csaba Memorial Laboratory of Bioseparation Sciences. And some of the old folks here, I hope remember him. The new ones, the younger ones, at least will know that he was the inventor of HPLC. Thank you very much.